Enzymes. Cofactors. Many enzymes require the incorporation of a non-protein molecule to become active. These molecules, called cofactors, can be metal cations like zinc, iron, or small organic groups called coenzymes. Most coenzymes cannot be synthesized by the body and are obtained from our diet as vitamin derivatives. Cofactors that bind to enzymes by strong covalent bonds are called prosthetic groups. Overview of cellular respiration. Photosynthesis converts the energy of the sun into chemical energy of bonds in compounds, such as glucose. Respiration involves the conversion of the chemical energy in these bonds into usable energy needed to drive the processes of living cells. Carbohydrates and fats are favored fuel components in living cells. As hydrogen is removed, bond energy is made available. The carbon-hydrogen bond is energy-rich. In fact, compared with other bonds, it's capable of releasing the largest amount of energy per mole. In contrast, carbon dioxide contains little usable energy. It is the stable, energy-exhausted end product of respiration. During respiration, high-energy hydrogen atoms are removed from organic molecules. This is called dehydrogenation and is oxidation reaction. The subsequent acceptance of hydrogen by hydrogen acceptor, oxygen in the final step, is the reduction component of the redox reaction. Energy released by this reduction is used to form a high-energy phosphate bond in ATP. Although the initial oxidation step requires an energy input, the net result of the redox reaction is energy production. If all of this energy were released in a single step, little could be harnessed. Instead, the reductions occur in a series of steps called the electron transport chain. Glucose catabolism. The degradative oxidation of glucose occurs in two stages: glycolysis and cellular respiration. Glycolysis. The first stage of glucose catabolism in glycolysis is glycolysis. Glycolysis is a series of reactions that lead to the oxidative breakdown of glucose into two molecules of pyruvate and the production of ATP, and the reduction of NAD+ into NADH. All of these reactions occur in the cytoplasm and are mediated by specific enzymes. Glycolytic pathway. Note that at step 4, fructose 1,6-diphosphate is split into two 3-carbon molecules. From one molecule of glucose, a 6-carbon molecule, 2 molecules of pyruvate, a 3-carbon molecule, are obtained. During this sequence of reactions, 2 ATP are used and 4 are generated. Thus, there is a net production of 2 ATP per glucose molecule. This type of phosphorylation is called substrate-level phosphorylation. Since ATP synthesis is directly coupled with the degradation of glucose without the participation of an intermediate molecule such as NAD+. 1 NADPH is produced per pGal for a total of 2 NADH per glucose. The net reaction for glycolysis is as follows: 1 glucose, 2 ADP, 2 phosphorus, 2 NAD+, results in 2 pyruvate, 2 ACP, 2 NADH, 2 hydrogen ions, and 2 water molecules. At this stage, much of the initial energy stored in the glucose molecule has not been released and is still present in the chemical bonds of pyruvate. Depending on the capabilities of the organism, pyruvate degradation can proceed in one of two directions. Under anaerobic conditions, in the absence of oxygen, pyruvate is reduced during the process of fermentation. Under aerobic conditions, in the presence of oxygen, pyruvate is further oxidized during cellular respiration in the mitochondria. At fermentation, NAD+ must be regenerated for glycolysis to continue in the absence of oxygen. This is accomplished by reducing pyruvate into ethanol or lactic acid. Fermentation refers to all of the reactions involved in this process. Glycolysis and the additional steps leading to the formation of ethanol or lactic acid. Fermentation produces only 2 ATP per glucose molecule. Alcohol fermentation. Alcohol fermentation commonly occurs only in yeast and some bacteria. The pyruvate produced in glycolysis is converted to ethanol. In this way, NAD+ is regenerated and glycolysis can continue. Lactic acid fermentation. Lactic acid fermentation occurs in certain fungi and bacteria, and in human muscle cells during strenuous activity. When the oxygen supply to muscle cells lags behind the rate of glucose catabolism, the pyruvate generated is reduced to lactic acid. As in alcohol fermentation, the NAD+ used in step 5 of glycolysis is regenerated when pyruvate is reduced.